monocarpellary. So mono means single, carpel means nothing but pistil. So hilum, it is the region where the body of the ovule fuses with the funicle. There are 3 cells at the micropylar end, wherein one cell is the egg cell and the other 2 cells are called as the synergids. Hello everyone, welcome to today's session, which is a continuity of the session 1, that is sexual reproduction in flowering plants. I am Dr. Divya, Biology Faculty, Vidhyaashram PU College, Mysuru. So in the previous session, we had studied about the pre-fertilization structures, wherein we had mainly concentrated on the male reproductive structure, that is the stamen, the microsporangium and the pollen grain that are involved in bringing about the pre-fertilization events. So in this particular session, we will concentrate on the female reproductive structure, which also plays a major role in bringing about the pre-fertilization events. Female reproductive structure, it is called as the pistil or the gynecium. So the gynecium, it is the female reproductive part of the flower. So when you look at the gynecium, it is made up of stigma. Then you have the style. So remember, in the male reproductive structure, we had the stamens. So in the stamen, you had the anther and you had the filament. But in this case, since we are talking about the female reproductive structure, which is called as the gynecium or the pistil, it consists of the stigma, it consists of the style, and it is made up of the ovary. So when you look at different flowers of different species, you will find that there is a great variation in the arrangement of the female reproductive structure, which is the pistil or the gynecium. So you can find that in some of the flowers, gynecium, it is monocarpellary. So mono means single, carpel means nothing but pistil. So if the flower has one carpel or one pistil, it is called as monocarpellary. And one more, if it has more than one pistil, it is called as multicarpellary. So multi means more than one, many in number. So in a flower, you can find that the female reproductive structure, which is the pistil or the gynecium, can either be monocarpellary or it can be multicarpellary. So monocarpellary means it has only one carpel. Multicarpellary means it has more than one carpel or the pistil. So you can see here, this is multicarpellary. In the image, I have put photographs of multicarpellary gynecium or multicarpellary pistil. So you can see here, there are numerous amounts of the female reproductive part being present, that is the gynecium. So that is why it is called as multicarpellary. So if it is multicarpellary or if more than one pistil is being present, then they can be either syncarpous or they can be apocarpous. Syncarpous means in synchrony, in synchronization, sync, they are synced. So that is why means they are fused. So carpus is nothing but carpel. So the fused carpels, if the pistil, they are fused together, you can call it as the syncarpus. And if they are free, that is, if more than one pistil is present and if they are not connected to each other, or if they are not fused with each other, or if they are not sticking with each other, they are separate, then you can call it as apocarpus. So This condition you can see only in multicarpellary Gynecium. So in the case of multicarpellary Gynecium or multicarpellary pistil, there are two forms that occur. One is, it can either be syncarpous, that is the pistil or the Gynecium can be fused, or they can be free, that is apocarpous. So you can see here, I told you multicarpellary, they have more than one carpus. So in this case, you can see that all the carpels or the pistil, they come and fuse at one particular point and it continues like that. That is why it is called as syncarpous. So it is called as syncarpous. So why is it called syncarpous? Because it is multicarpellary and all the carpels come and fuse at a point together up till the ovary. It is continued. So it is clear in this particular diagram. So you can see that it is multicarpellary. It is made up of here more than one pistil, and all these pistils, they come together to join at one particular region. That is why it is called as syncarpous. All the pistils, they are fused at one particular region. But in this case, I have put the flower of magnolia, which is champaka flower. So here you can see that there are different carpels. So here also it is multicarpellary, but they are free. They are not fused. Everything is not fused. They are free. That is why it is apocarpous. So the Gynecium, it is the female reproductive part of the flower. It can be either monocarpellary, wherein a single pistil may be present, or it can be multicarpellary, wherein more than one pistil is present. If it is multicarpellary, two forms can occur. One is either it can be syncarpous, wherein the carpels or the pistils are fused together, or they can be apocarpous, wherein the carpels and the pistils are free. They are not fused, they are free. So the pistil or the Gynecium, as I told you, it has three parts. One is the stigma, the second part is the style, and the third one is the ovary. So we'll study about these parts or structures one by one. So moving on further, in the pistil or the female reproductive part, that is the Gynecium, the first structure that you observe is the stigma. So stigma is very important because when pollination occurs, when the pollen grains fall on the stigma, it acts like a bed for the pollen grains. So the stigma is sticky, or it will be hairy. So the pollen grains can stick on the stigma. So therefore, the stigma is the part where the pollen grain lands on a flower. So stigma, it is the region where the pollen grains will travel from another flower and it will come and place itself or locate itself in that particular position. So next important part, it is the style. So style. So similarly, like how in an anther you had the filament, in the Gynecium or the pistil, you have the style. So style, it is long and a slender part, and to this style, the stigma is being attached at the tip. So one end of the style is attached to the stigma and the other end, it is attached to the ovary. So the next important part in a Gynecium or in a pistil is the ovary. So you can see here, even this female reproductive part also, it is placed on the thalamus. So the first important part of the Gynecium, it's the stigma. Stigma is usually hairy and sticky. Why? Because so that it can collect the pollens, the pollen grains. When it falls on the stigma, it should not fall off. So that is why it is sticky. So therefore, stigma, it is the part or the region where the pollen grains come and sit on it or land on it. So next important part, it is the style. So style, it is long and slender and it is attached to the stigma. So the third important part, it is the ovary. So when you look at the ovary, you can see that it is bulged and it is the basal part. So one end of the style, it is connected to the ovary. So the ovary, it is bulged, it is somewhat swollen, it is bulged and it is present, it is the basal part because it is present at the base of the stigma or the style. So it is the basal part and inside the ovary, you can see the ovules being present. So the ovary, it is the bulged basal part of the stigma and inside the ovary, you can see the ovules being present. So when you see the ovules or when you take a cross section of the ovary, you can see locules or cavity being present. So you have the cavity, that is the ovarian cavity, and you have locules like this, locules, and then you have inside the locule, you will have the placenta. So you can see here clearly, this is the placenta. So say for example, you have an ovary like this with two locules. So this is the locule, it's the locule, then you have the ovule and the ovule is connected by a placenta. So this is the placenta. So the ovary, it is the bulged basal part of the pistil. Inside the ovary, you can find the ovules and inside the ovarian cavity or inside the cavity of the ovary, you can find the locule, that is usually 2 or more than that locules will be present and inside the locule is the placenta. So you have the locules and inside the locule you have the placenta and when you see the placenta, it's a connectivity between the ovules. So the placenta, it arises the ovule. So the placenta is the one which holds the ovule inside the ovary in its place. So this ovule is also called as the megasporangia. So when you see a ovule, it is also called as the megasporangia. So remember in your previous session, you had studied about microsporangia, wherein you had the pollen grain. So the pollen grain is called as the microsporangia, right? But in the female gametophyte, the ovule is called as the megasporangia, which plays a very important role in bringing about reproduction because this is the ovule that forms the seed in order for the plant to continue its next generation. So now let us study about the structure of the megasporangia. So what is this megasporangia? It is nothing but the ovule. So where is the ovule being present? Inside the ovary. So let us study about the structure of the ovule, which is nothing but the megasporangia. So the ovule, it has the following parts. So we'll study about each of the parts one by one. So the first part, it is the funicle. So this is the funicle. So the funicle, it is a stalk-like structure. So this is the stalk, it looks like a stalk. So that is why it is a stalk-like structure. So the funicle is a stalk-like structure and it helps in attachment of the ovule to the placenta. So as I told you, you have the ovary. Inside the ovary, you have the placenta and you have the ovule. So funicle is the one which helps in attaching the ovule to the placenta. So therefore it is called as the stalk of the ovule. So funicle, it is the stalk of the ovule which helps the ovule in attaching itself to the placenta. So that was about the funicle. So next we'll study about the hilum. So hilum, it is the one or it is the structure or you can also call it as the junction which forms a connectivity between the ovule and the funicle. So the region where the funicle attaches itself to the ovule, it is called as the hilum. So this is the hilum. So this is the junction. So funicle, it is the stalk of the ovule. The funicle helps the ovule to attach or connect itself to the placenta. Next part is the hilum. The hilum, it is the region where the ovule fuses with the funicle. So you can see here the entire ovule, it fuses with the funicle, and this particular junction where the fusion occurs, it is called as the hilum. So we'll move on to the next part, that is the integuments. So any structure in a flowering plant has to have a protection. Why? Because flower is the main reproductive organ of a plant. It's not the leaf, nor the stem or something else. It is the flower. So here, when we think of the flower, the ovules are the one which play a very, very important role in reproduction or bringing about sexual reproduction in a flowering plant. So the ovule has to be protected. So here also the ovule, it has integuments. Integuments are nothing but coverings or hard layers or hard coverings. They are called as the integuments. So the ovule is protected by the integuments. So the integuments, they act as a protective envelope. Envelope is nothing but to cover. They act as a protective envelope and they cover the entire ovule. So there is one more thing which you have to keep in mind here. Though the entire ovule is being covered, there is a region in the ovule which is not covered by the integument. It is called as the micropyle. So first I'll tell you what a micropyle is and then we'll move on to the integument. So can you see here, there is a small opening here. Why there is an opening? Because the integuments. So these are the integuments. The integuments, they are not covered here. It is incomplete covering here. So though the entire ovule is being covered by the integuments, there is a small region where the integument doesn't cover the ovule. That particular region is called as the micropyle. Micropyle, a small opening, it is called as the micropyle. So integuments, it is the protective envelope that covers the entire ovule, and you have one more structure called the micropyle. So the micropyle, it is a small opening in the ovule which is not covered. So it is not covered by the integument. So the region where the integuments doesn't cover the ovule, there is a small opening or a gap. That particular region, it's called as the micropyle. So this is the micropyle. And also you have to remember that the ovule has two poles. This is one pole and this is the other pole. So here one end of the ovule, it is the micropylar end. It is the micropylar end, and the other end, it is the chalazal end. Chalazal end. So you have the micropylar end. So this tip is the micropylar end, wherein you have the micropyle. Micropyle is nothing but micro, a small, something that is small, pile, an opening. So micropyle, where there is an opening. Why is there an opening? Because it is not covered by the integuments, or it is not covered by any of the layers. So that is why. And next, one more end, you have the chalazal end. This is the chalazal end. So two polar ends. An ovule has two polar ends. One is the micropylar end and the other one is the chalazal end. So we'll talk about the chalaza now. So chalaza is the basal part of the ovule. So chalaza, it is the basal part of the ovule and it is present opposite to the micropylar end. So you have the micropylar end here. So this is the micropylar end, and opposite to the micropylar end, you have the chalazal end. So this is the chalazal end. So chalaza, it is the basal part of the ovule. This is the upper part of the ovule. This is the basal part of the ovule. It is the basal part of the ovule that is present exactly opposite to the micropylar end. So that is about the chalaza. So next we'll move on to the nucellus. So nucellus, it is present inside the integuments. So let us brush up. What are the integuments? Integuments are the layers or the thick hard envelopes that cover the ovule. So inside the integuments, you have the nucellus. So in nucellus, it is a group of cells or a mass of cells that is present inside or within the integuments. So what does this nucellus do? They have a particular function. They nourish the developing embryo. So you have the nucellus. So this particular nucellus nourishes the developing embryo. So where is this embryo present? It is present inside the embryo sac. So the nucellus, it is a group of cells that is present inside or towards the inside of the integuments, and the main function of the nucellus is whatever embryo, that is during the pre-fertilization events, there is embryo development. So which we'll study in detail in the coming session when we study about the pre-fertilization events. So after the pre-fertilization events, when the embryo develops, the embryo has to get nourishment for its further growth, right? So from where is the nourishment got? It is got from the nucellus. So the nucellus, it has abundant or it has a large, it is a storehouse of various food materials, maybe in the form of sugar, saccharides, etc. And it helps in providing nourishment to the developing embryo. So the nucellus, they nourish the developing embryo. So where is this embryo present? It is present inside the embryo sac. So the embryo is usually formed after the pre-fertilization event. So it is present in the embryo sac. So inside the embryo sac, you have the embryo. So embryo sac, it is actually the female gametophyte. So in the earlier session, you had studied that pollen grain is the male gametophyte, right? Here, embryo sac is the female gametophyte. Once the pollen grain, it enters into the female reproductive structure, all the events will occur in the female gametophyte or the embryo sac. So we will study in detail about the female gametophyte, which is also called as the embryo sac. So inside the embryo sac, what all are the structures that are present? We will study. Why is it important? Is because if you need to understand or if you need to clearly know about or learn about what are the pre-fertilization events that occurs inside the female gametophyte, then it is important for us to understand first the structure of the female gametophyte or the embryo sac. So I told you the ovule has a micropylar end and a chalazal end. So this is the inverted form. So you have the micropylar end and the chalazal. Do not get confused. Towards whichever side the egg apparatus is present, it is always the micropylar end, and towards whichever side the antipodal cells are present, that will always be the chalazal end. Since this is in an inverted form, I have written the chalazal end at the topmost corner and the micropylar end at the bottom corner. So you have in an ovule, you have the chalazal end and the micropylar end. In the micropylar end, you have the egg apparatus. So in the egg apparatus, you have 3 cells. So 1, 2 and 3. So 3 cells are being present. So you have 3 cells. Remember, I had told you the embryo sac, it is 8 nucleate. But in spite of being 8 nucleate, it has 7 cells. So we are discussing why is it so? It has 7 cells, though it is 8 nucleate. So in the micropylar end, the embryo sac is made up of the egg apparatus, and the egg apparatus consists of 3 cells, and out of the 3 cells, the one is called as the egg cell. The one it is called as the egg cell, and the other 2 cells are called as the synergids. So one is called as the egg cell, and the other 2 cells it is called as the synergids. It is called as the synergid. So now you have 3 cells here, and each cell has its own nucleus. So now at present, how many cells are there? There are 3 cells at the micropylar end, wherein one cell is the egg cell and the other 2 cells are called as the synergids. And next we have the antipodals. So these antipodals, they are present at the chalazal end. Again here, all these 3 cells at the chalazal end, you call it as the antipodals. Now together, how many cells are over now? 1, 2, 3, 4, 5, 6 cells, each cell having 1 nucleus. So 3 cells, totally 6 cells, totally 6 nucleus, right? So here the chalazal end has 3 antipodals and the micropylar end has 2 synergids and 1 egg cell. So altogether we have 6 cells and 6 nucleus. So it is 6 celled, 6 nucleate, right? So next we'll move on to the central cell. So central cell, it is present at the center. So this is the central cell. Central cell. So it is present at the center of the embryo sac, and it has 2 polar nuclei. So here 2 polar nuclei are there. So here 2 polar nuclei are there, but where is it present? It is present inside one cell, which is called as the central cell. That is why the embryo sac, though it has 8 nucleus, it is 7 celled. How many cells you have now? 3 cells at the micropylar end, 3 cells at the micropylar end, 3 cells at the chalazal end, and 1 at the center. Right? It is 7 celled. But why is it 8 nucleate? Because 3 cells having 3 nucleus each at the micropylar end, and 3 cells having nucleus each at the chalazal end, and in the central cell you have 2 nucleus. That is why it is called as 8 nucleate. So that is why, in spite of the embryo sac being 8 nucleate, it is a 7-celled structure. So this is about the female reproductive structure, wherein you studied about the stigma, you studied about the style, and you studied about the ovary. So inside the ovary, you studied about the placenta, then you studied about the ovule, and in the ovule, you came to know that the ovule has 2 poles, that is the micropylar end and the chalazal end. At the micropylar end, you have a stalk-like structure which is called as the funicle, and the region where the funicle attaches itself to the ovule, that or the placenta, that particular region you call it as the hilum, and after that the ovule, it is covered by 3 integuments, and at the micropylar end, a small region of the micropylar end, it is not covered by any of the integument. There is a small opening, that is why it is called as the micropyle, and at the basal part of the ovule, that is exactly opposite to the micropyle, you have the chalazal end. So inside the ovule, you have the embryo sac. So what are the structures that are present inside the embryo sac? And remember, the embryo sac, though it is 8 nucleate, it is called as 7-celled. So at the micropylar end, you have 3 cells, wherein one is the egg cell and the other 2 are the synergids. And in the chalazal end, you have 3 cells again, which is called as the antipodal cells. And in the center, you have a central cell, which consists of 2 nuclei or 2 polar nuclei. So this is about the female gametophyte. I hope you have understood this session well. So in the next session, we shall study about what are the events that occur during the pre-fertilization process, and in the coming session onwards, we'll also study about what are the events that occurs in the double fertilization as well as the post-fertilization. So in the next session, we will concentrate on the pre-fertilization events, wherein we'll study about the microsporogenesis. Microsporogenesis is brought about by the microspores or the pollen grains, which is related to the male gametophyte. And then we'll study about the megasporogenesis, wherein we'll learn about the formation of the embryo sac. So you have studied in the embryo sac or the female gametophyte what are the structures that are present. In detail, what all events will take place inside the embryo sac, we'll study in the next session. So I hope you have understood this session well. Thank you.